Howdy, friends. Today we're going to take a look at protein synthesis and codons practice. I had assigned some of this last week, and I just want to go over some of this again to make sure that everybody is clear on this. So I'm going to start here by reading this. Protein synthesis is the process where a sequence of DNA is used to build a protein from individual amino acids. The first step in this process is called transcription, where a coding region of DNA is converted to messenger RNA or mRNA. During transcription, messenger RNA is made from the DNA sequence following the base pair rule. Remember, that's A goes with T, C goes with G, except RNA does not contain the base thymine, but instead has uracil right there. The mRNA then leaves the nucleus and goes to a ribosome in the cell's cytoplasm. The ribosome reads the message 3 bases at a time, and that's called a codon. So those 3 bases together, AUG, CUG, CCC, whatever it is, that's called a codon. Each codon will specify a single amino acid. The amino acids are then joined together and folded into a protein, and this process is called translation. All right, so real quick, key points. DNA is used to make a copy of messenger RNA. That's called transcription. Messenger RNA leaves the nucleus and goes to the ribosome. Remember way back at the beginning of the year, I said you got to know that ribosomes make protein? There you go. 3 bases equals a codon. 1 codon equals a single amino acid. A chain of amino acids equals a protein. And protein synthesis is also called translation. All right, let me scroll down here. Whoops. Operator error. Hold on. Oh, there. All right, got that figured out. So biologists use a codon chart or a codon wheel to determine the amino acids. Amino acids are usually abbreviated on these charts as 3-letter words like cis and ser. So this is the codon chart. This is the codon wheel. They tell you the exact same thing, just in a different form. So for example, let's say that I want to figure out what GCA would be on the codon wheel. I would come over here to G, then go to C, then go to A, and that looks like ALS, right? So let's scroll down here a little bit and we will do some practice. All right, so here are our practice problems. We start with C, C, C. This is an easy one to start with. We're going to come up here to C, right? We always start here in the middle. And then we go to C, which is here. And then we go to C here, and that's going to be PRO. So the amino acid that I'm looking for is going to be PRO. Okay? Let's try the second one together, and then I'm going to make you do the rest. We have AGU. We're going to start here in the middle at A. We're going to go to G, and then to U, and that is SER. So again, you're going to start in the middle and work outward. Okay? All right, for the second part, write the codon that corresponds with each amino acid. There may be more than one. The full names are written, but the codon chart only shows the first 3 letters. So let's start with proline or proline. Okay, I'm going to come back up to the chart here, and I have to find proline. That's the hard part. Okay, I see it right here, PRO. Okay, it looks like if we look carefully, it could be any of these four for the last number, but the first one is going to be C, C, and then it could be A, U, G, or C. So for my answer down here for proline, I'm going to write CCG. I could have written CCU, CCC, CCCC, CCU, CCA, or CCG. I just randomly chose G, okay? And that translates to proline. So again, we have to find the name, but remember that we always start in the middle, okay? Another way we could do that is by looking at the codon chart here. And this one is a little bit tougher, but here's pro right here, and it actually gives us all of those answers right there. So I could have done it that way, okay? Moving on, this we are looking for the start codon. I want to know what those three bases are, and if we come back up to our wheel or our chart, you can see that start is right here, and start is right here, and that is a very distinct codon, AUG or AUG, okay? There's also a stop codon. There are actually several over here, so you can look at those as well. The last thing I want to go over before I set you guys loose is for each sequence of DNA, you need to write the complementary RNA sequence and then use the codon chart to determine the amino acid. So RNA, let's see, opposite of T is going to be A. Opposite of A is U. Opposite of C is G. Okay. C is going to be G. Opposite of A, U. And opposite of T, A. Okay, etc. You guys kind of know how this works now, right? Now, but the next thing that I'm asking you to do is then to take that codon and transfer it into the proper amino acid. So if I'm looking for AUG, I'm going to come back up to the top. I'm going to say AUG. Oh, look, it's MET, or the start codon. So down here, I would write MET. Oops. All right, and then we'll do this next one together. GUA. GUA is valine. So right here, BAL. Okay, I hope that makes sense. I am assigning this very document that I am writing in right now for you to complete. You can write directly into the document. If you have questions, please email me. In the meantime, feel free to use this document, stop it, play it back, etc., and let me know if you have any problems. Bye, guys.